of prokaryotic genomes.
Okay, so thank you everyone for being here. And yes, I'll talk about the prokaryotic genomes, about how they are non-uniform, and we will talk about empirical analysis of actual genome data and some more mathematical modeling of the genome structure. Okay, and now I cannot advance about this. No.
It might have been clicked on a different window. Just click the mouse over the presenter mode, and then you should be
There. Is that good? Okay.
Okay, so the fact that microbial genomes are non-random spatially and non-static temporally was known for many decades. You can see that on the very first bacterial chromosome map compiled in 1964, you can see that genes with similar names are clustered closely together, like whatever MET-BF, ARG, ACFH, and it means it suggests that the spatial structure of bacterial chromosomes is non-random. And a few decades later, when we had first few completely sequenced genomes, their comparison helped to realize that there is a lot of dynamics in prokaryotic genomes. Genes come and go, and not all bacterial chromosomes have the same complement of genes. And then it took a few more decades to realize that these two approaches to microbial chromosome structure, prokaryotic chromosome structure, can and should be combined into spatial temporal analysis. Both mutual arrangement of genes and the dynamics of that could be studied simultaneously, and this provides insights which are inaccessible if you are looking from only one perspective. So we have published already several papers on using this spatial temporal approach to microbial genome dynamics. Some other works are in progress, but here I want to pay attention to this work explores the gap about on the scale. If you go from single fine-grained single nucleotide to the whole genome site, we know a lot about dynamics on single nucleotide level. We know a lot about evolution of protein domains, operons, and systems on the scale of few kb to 10 kb has been studied. We are now looking at the sub-chromosome scales from roughly 30 to 300 kb. So this is necessarily very brief overview of what we are doing. We take circular chromosomes and we tag genes on chromosomes, 1 or 0. We can use, in principle, whatever binary traits we want. In this particular case, we are looking at genes we infer to be ancient or young, relatively, of course, but we can map them on a chromosome, and then we use some computational tricks to find how uniform or how non-uniform the distribution is on the chromosome. So we build cumulative profiles along the chromosome. We do skew profiles, subtracting the average density from each position. We smooth it. We find peaks and valleys on this distribution, and it gives us the regions with higher than average and lower than average density of tags. Then we do maximum likelihood adjustment of boundaries, and based on archaic information criteria, we drop some segments which are not informative. After this, we end up in chromosome partitioned into high density and low density segments with respect to tagged genes. Very, very general overview. Now, about the tags we are using here. If you, this is a very sort of well-known phenomenon that whatever group of genomes, not necessarily microbial genomes, any eukaryotes, large viruses, if you look at the distribution of genes by the number of genomes or fraction of genomes they are present, they get you this U-shaped curves. You have a lot of singletons or very rarely distributed genes, and you have a number of genes which are present in the majority of the group, so the fraction of genomes near 1, with some generally with a lower bottom of the U. In between, of course, depending on the specific group you are talking about, there could be peaks and valleys there. So what we do here, we take genus, formally taxonomically defined genus of microbes. We cluster all protein sequences encoded in those genomes using similarity threshold 0.5. We build those commonality curves. From that, we build the distribution. Now, this curve is by gene families, and in our particular case, by clusters. Then we map them back to genomes, build the distribution of commonalities across the genomes, and then just purely formally define the bottom 1/3 of the genome of genes as young, and the top 1/3 as ancient, again, relative to the particular genus. So these definitions would be different between genera. And then map them to chromosomes, and then apply the computational pipeline I briefly discovered. This is what we get. We see that with very, very few exceptions, the distribution of these genes is very uneven. Almost all chromosomes, and the exception are very small genomes, very reduced genomes of intracellular parasites. They very clearly break down into regions of high density and low density, relatively high density, relatively low density, and the inner circles show mapping of the young genes onto chromosomes, and outer circles show mapping of ancient genes on the chromosomes. You clearly see that the distribution is non-uniform. You have regions enriched and depleted with young and ancient genes, respectively. And here you show examples across different bacteria and archaea, and here are like more formal analysis of these distributions. We have 175 genera. You see that for overwhelming majority of genera, these are cumulative distribution functions. You have highly significant segmentation. You have typically relative enrichment between 2 and 4. So the high density regions have 2, 3, 4 times as many genes, ancient for example, genes, then depleted regions, the mean length of segments has a remarkably narrow distribution. If you would do some random experiments, would have them all over the place. Actually, like you have whatever 3 binary orders, so maybe a factor of 8, and that typically much longer segments are formed by ancient genes than by young genes. And also the segments are fuzzy. They are not like 100% versus 0%. The typical density of enriched segments is around 0.5, 0.6, and for depleted segments is by a factor of 2 to 4 less. So as an intermediate summary, if we stop here and do nothing more, what we know. All archaea and bacteria show strong tendency for uneven distribution, except for what I said about intracellular parasites. Other than that, there is no taxonomic signal. It is roughly the same across the board between bacteria and archaea, between different phyla, different genera. Typical lengths of segments is in low hundreds of kilobases, so low hundreds of genes. You have practically no correlation between genome size and the length of the segments, which means, of course, that the number of segments strongly correlate with the genome size. So again, these genome chromosome segments, they are of similar size, and you have as many as you need to cover the chromosome. There is a enrichment by several fold, but they are fuzzy, so they are typically below 70% enriched. From this, you can immediately understand what these segments are not. They are not operons. They are not what they call genomic islands. So like we know that there are pathogenicity islands, integrated prophages, defense islands. They're typically longer than that. These segments do not follow GC-SKU domains. In bacteria, there are typically 2 of them, GC enriched and GC depleted. So this does not, and with archaea it's more complex, but anyway, it is not symmetrical around the origin, so it is most likely not an effect of lagging strand. If you look at alternative definition of target genes, you can, for example, like here, look at E. coli genome and map genes mapped predicted to be in last bacterial common ancestor or in last common ancestor of proteobacteria, or you can look at genes with lethal knockout phenotype. They all form segments, and you can see by chi-square p-values that the segment and enrichment value segmentation is stronger from left to right. So the more immediate measure of the gene importance, like last bacterial common ancestor is whatever what was conserved in 4 billion years, maybe 3 billion years, and here we are talking about what is lethal in this particular strain of E. coli. So you have stronger signal. We tried to compare this with 3D chromatin structure, which is known for a few bacteria. It might look like we do somewhat match the one in archaea, but totally discordant relative to bacteria, so this is not it. You can look at comparison of genomes at different density, at different evolutionary distances. You see that the rearrangement start in local points, and then you lose synteny. You lose synteny, but you don't lose segmentation. So this is not about the protected regions protected from recombination. This is about biased movement. So we did make a model and run simulation model based on the two features, that some genes are less mobile than other genes, and some sites on the chromosome serve as an attractor for gene translocation. And funny enough, you can get from this model the simulated chromosome structure, which is very similar to what we observe. This is a particular measure of chromosome structure, and don't have time to go into details. Multi-scale complexity profiles and the peaks and valleys on these profiles tell you about the characteristic structural scales. So the simulated data actually pretty closely follow the real one. This is one example we have it run for everything, and this is what we see. So conclusions. We do have a strong segmentation, very non-uniform distribution, segments of variable but narrowly distributed sites, and we seem to have some, not evidence, but some suggestions that this structure is dependent on differential mobility and differential attractions of sites on the chromosome. This work was done as a collaboration between our group at NCBI and people from Radboud University in Netherlands. Thank you.
So we have about 2 minutes for questions. And since nobody's coming up to ask a question, you go first.
Yeah, thank you for a very interesting talk. I have actually a couple of questions. First, I didn't quite understand what are the assumptions of your model. And second question, you showed the consistency between your sort of chromosome organization with 3D map. And you see that only consistency for one organism out of 3. Can you please comment on that, please?
Okay, so people working, second question first. People working with Archaea noticed that important, quote unquote, important genes, those which are higher expressed, those which are present in wider range of Archaea, they typically concentrate in segment B. Segment A on 3D structure of sulfolobus chromosomes, and less important genes in segment B. And this is, if you can see these light blue segments, they are more or less similar. But if, for example, you look at E. coli, there is nothing similar. So our conclusion is that it is not chromatin structure that drives the differential distribution, it's the other way around. Placement of localization of those important genes, in particular chromosome locations, drives the maintenance of 3D chromatin structure, at least in Archaea, apparently not in bacteria. Now, the second question about the assumption of the model, single gene translocations, 2 classes of genes, high and low mobility, and attractors in the vicinity of each you are more likely to translocate than elsewhere. And this is enough to get this structure, and by varying the strength of the attractor and the differential mobility, you can fit quite well to the observed structure.